Hello everyone. This is PHN 313 video presentation on biomedical signal and image processing. I am Ashutosh Kumar, BTech third year engineering physics, and let's start with the presentation. So biomedical signals, any signal that can be classified into a biological if it's recorded from a living system, let's say mostly a human body, and conveys information about the state or behavior of that system. For example, if we take a thermometer and take the temperature reading of a patient, you must have seen the X-ray scans, the CT scan, the ultrasound results, the MRI scan. These are all uh, made from or constituted from the biomedical signals. Now, these can be either one-dimensional if they depend on a single variable, let's say time, or it can be a multi-dimensional if they depend on several variables such as spatial coordinates. Like if we take biomedical images, they are multi-dimensional signals. They contains both spatial coordinates and vary with time also. So the biomedical signal processing, it includes the analysis of the measurements like heart rate, blood pressure, oxygen saturation levels, blood glucose, nerve conduction, and many more medical parameters. In order to provide useful information upon which the doctors or researchers can make decision based on the symptoms, and these data can also help in multiple ways to process these signals using a variety of mathematical formula and algorithms, which we will be seeing later in this PPT. Now, the purpose of the signal processing is to selectively eliminate the irrelevant information from a signal, such as the noise or any sampling or something, so as to make the information of interest more easily accessible to a human observer or a computer system. The reason for this is that it is never possible that you will be adding some information to a biomedical signal. You can also be refining using some filters, or you can just channel any information from a biomedical signal as per your utility. Now, in a typical biomedical application, signal processing may include four stages. First one is data acquisition, next is signal conditioning, then comes the feature extraction, and finally the decision making, like what we can do with this image and the data available. Now here, the goal of data acquisition is to capture the signal and encode it in a form that will be suitable for computer processing. And because the main concern here is we can't afford to lose information about any signal. We just have to filter out carefully using different tools. It typically consists of three stages, the data acquisition part. First one is transduction. It's as it is literal enough. This is the conversion from one form of energy to another. The signals need to be converted to analog voltages whose waveforms are ideally the same as those of the original signals. Then comes the analog conditioning that usually consists of amplifying and enhancing and filtering the analog signal measured with a transducer. And then come the last part of analog to digital conversion. It's quite plausible enough that it's a device that transforms a continuous time signal measured with a transducer with a digital signal that can be represented in a computer. Now, ADC are implemented as integrated circuits commercially as well as in for research purposes as well, and find multiple application in digital image processing, music recording, scientific instruments, encoders, and even display devices such as LCD. Now, conceptually, these analog to digital conversions can be divided into a series of two operations. First, one is sampling, in which any continuous time signal is converted into one that can be defined for discrete time functions. But the amplitude in these cases can take arbitrary values, but the time function will be discrete only. And the next one is quantization part, in which a continuous amplitude signal is converted into a digital signal that can only take a finite set of values. Now here the goal of signal conditioning, which is the third part, is to eliminate or reduce extraneous components such as the noise from the signal. Often this is done using linear filters, sometimes in combination with nonlinear operators too. The third part is feature extraction, means identifying and measuring a small number of parameters or features that best characterize the information of interest in a signal. The distinction between feature extraction and signal conditioning is not a rigid one, because in both we selectively eliminate irrelevant information. Then comes the part from analog to digital conversion, which is the very important concept in biomedical signal processing or any type of signal processing, is the sampling of a continuous signal. Because here we need discrete time signals, and that were only can be obtained from any continuous time source by sampling at regular intervals. This is the expression for sampling, and here Ts is the sampling interval, and Fs, which is 1 by Ts, is the sampling frequency, or we can say the sampling rate. Now here, what's the purpose is that we need to allow their processing by digital computers, and they take in discrete time signal function only. This is useful only if the information carried by the original signal is retained. Now retention of all the information during sampling is too important, because we can't compromise the information or data just for the sake of digital image processing or digital signal processing. Now, it's an important concept in there is Nyquist sampling theorem. It states that the signal xt, x as a function of time, contains no frequency components higher than w. W is some frequency, and it can be exactly reconstructed from samples taken at a frequency f is greater than 2 into w. Now, from Nyquist sampling theorem, we also get explicit interpolation formula for reconstructing our signal as a function of time from the discrete sampling. So inverse processes also can happen. This is the expression for that. Now let's come to we got to the data acquisition part. Now let's see how is the biomedical data classification. Now, due to high influx of medical data, manually classifying a large volume of data is no longer feasible. It is desirable to have automatic algorithms to efficiently and effectively classify the data on behalf of domain experts. Now here, the application of machine learning, deep learning, and artificial intelligence is inevitable into efficiently classify the data, and a reliable classifier avoids bias induced by human intervention and yields consistent classification. As we say for biomedical data classification, here are some tools related to artificial neural networks and artificial intelligence in general. These are support vector machines, decision tree, k nearest neighbor, that is KNN, naive Bayes theorem based on the Bayes theorem of probability, dictionary learning methods. We'll be seeing one by one. So this is the support vector machines. It's a concept in neural networks. It is basically a supervised machine learning algorithm that can be used for both classification or regression challenges. In the SVM algorithm, we plot each data item as a point in n-dimensional space, where n is the number of features you have, with the value of each feature being the value of a particular coordinate. Now, these are in a sample, let's say n-dimensional space. Whatever coordinate you choose, that is the data point. Then we perform classification by finding the hyperplane that differentiates the two classes very well. As you can see in the figure also, these are two types of data that is plotted on an n-dimensional space, and then we have a hyperplane that differentiate between the two classes well. Now, if any data come onto this, we just compare the distance in which class does it lie using the hyperplane system. So it is effective in cases where the number of dimensions is greater than the number of samples. Next come the decision trees. It uses a flowchart like a tree structure to show the predictions that result from a series of feature-based splits. Now these are upside down, which means the root node is at the top and that the root is splitted into decision nodes, and it further is sub-decision nodes, and at the end we will have terminal, or known as the leaf nodes. So in decision tree we get something prediction like if you choose this way, you will get onto this, you will stumble upon this output. So using decision tree also data can be classified. Let's say the data classifier is trained and a new data has been inputted into the system, and it will follow the decision tree. Like if it goes there, it will end up in this. So according to that we will classify all the data. The third one is the k nearest neighbors. It stores all the available data, training data, and classifies a new data point based on the similarity. It trains the model in such a way that when there's a new data input, it can be easily classified into a category. Let us go through the pseudocode for this KNN system. So first we load the training data set, and next we will prepare our data by scaling missing value treatment and dimensionality reduction as required. Now let's say we find the optimal value for k and then predict a class value for new data. For doing that, firstly we will calculate the distance from the training data and the new data point. We'll calculate distance between training data and new data point as per our chosen distance metric in the graph. Now we will sort these distances in increasing order with corresponding training data. From the sorted list, select the top k rows and find the most frequent class from the chosen k row. This will be a predicted class. So naive Bayes and dictionary learning methods. Naive Bayes is a classification technique based on Bayes' theorem of probability with an assumption of independence among predictors. That is, be mutually exclusive. The classifier assumes that the presence of a particular feature in a class is unrelated to the presence of any other feature. So pxy is equal to py in presence of x multiplied by px upon py. That is the what is the prior possibility multiplied by likelihood upon evidence. That is basically the Bayes theorem. Now, dictionary learning methods is a branch of signal processing and machine learning that aims at finding a frame called dictionary in which some training data admits a sparse, not dense representation. So here are the results. That this data will form a dictionary of certain of their categories like no noise, no outliers, many small, and few large outliers, and anyone which is not dense, that will be framed into called a dictionary. Now let's come on to the biomedical image processing part. Image processing is any form of signal processing for which the input is an image and the output may either be an image or set of characteristics or parameter related to image. We will also see the representation of image in digital format. So biomedical image processing includes analysis, enhancements, and display of images captured via instruments such as X-ray, MRI, ultrasound, CT scanners, and other optical imaging technology. These images are composed of pixels to which discrete brightness and color values are assigned. The techniques were originally developed for analyzing remote sensing data that can be modified to analyze the outputs of medical imaging systems in order to assess the symptoms of medical conditions. Now, the multiple tasks performed in image processing are image enhancement, construction of 3D images from 2D, restoration, smoothing, color correction, contour detection, many more. We will be doing that using a software, ImageJ, for an OCT scan image later in the PPT. So the principles of biomedical image processing and what types of images we got. So an image is usually a function of two variables, example fxy, where f is denoted as the brightness of image in this space and xy is the location. It can also be defined as an array, a matrix of pixels arranged in rows and columns, and the image information is converted into an array of integers, and then image can be manipulated, processed, and further displayed at desired output. So basically there are two types of images, analog and digital. An analog image can be described by spatial distribution of brightness or gray levels, reflecting the distribution of the detected energy. Black and white images require only one gray level of intensity variable, while color images require multiple variables, namely RGB. The RGB intensities can produce any selected color at a spatial location of the image when combined together using different intensities. Now, a digital image is discrete in both spatial and intensity gray levels and a discrete spatial location of a finite size with a discrete gray level value. It's called a pixel. The dimension of the pixels are determined by spatial sampling of the image. Now, it's an important concept in image processing is the Fourier Transform. It's a linear transformation that provides information about the frequency spectrum of the signal. The Fourier Transform function ft is given by this expression. It is used in image processing for image enhancement, restoration, filtering, and feature extraction to help image interpretation and characterization. Basically, it decomposes a signal ft into frequencies that made it up and used in reconstruction methods for medical imaging systems. Once the image gets transformed into frequency domain, we filter out the degradation due to the noise in the signal and also the unwanted frequencies. This information helps to restore the image using inverse Fourier transformation given by this expression. Now, as we talked about, I have used a software called ImageJ for all the image processing part. So here's the image color adjustment. This is the original image provided to me by Dr. Mayank Goswami, who is the assistant professor at IIT Roorkee. And this is the adjusted image. You can see I have adjusted the brightness by 39 from 0, and you can see the changes. Next one, I enhanced the images. Like this is the original image as we talked about, and in the second image I have added some noise and enhanced by 2%. The original image is enhanced by 0.03 or 0.3%. And the third image is I have noise added, smoothened, and sharpened the image. This is the 3D construction of image. This is the 3D view of our the previous 2D image. And here it's the 3D rotation that I will play now. Here, let me see. In the image J3D viewer, I will rotate the image. Yes. This is the 3D representation of our image dataset. Okay, let's move on to next. So this is the Fourier transform of the image. As you can see, this is the original image and this is the Fourier transform. So you can see, you can now we will particularly filter out the frequencies, remove the noise for useful information from that Fourier transform of image dataset. So what are the components of biomedical image processing? First one is the image formation. It includes steps from capturing images to form a digital image matrix. The steps are image acquisition, the action which is known as the action of retrieving an unprocessed raw image from the source. That can be X-ray scan, MRI scan, or something like that. Digitization of the image acquired, raw image acquired. It's the process of converting image information into a digital format organized into discrete units of binary data. Analog to digital converters are being used in the process to convert the signal acquired from the instruments to digital format. Now the second one is image enhancement and visualization. It refers to all types of analysis done on the digital data acquired from the image. The purpose is to enhance the image in terms of color and contrast to better visualize the features of interest. The steps are, firstly, we will eliminate the noise from the image using linear filters, high and low pass filters. Then we adjust the different parameters of the image such as brightness, contrast, visibility, color, saturation, etc. The third component of this biomedical image processing is the image analysis. It basically includes all the steps of processing which are used for quantitative measurement and interpretation of biomedical images. Using the type and content of image, the data is being integrated into an algorithm on a high level of abstraction. Now the process of image analysis is very specific and developed algorithms can be transferred directly to application. For this, we have used different CNN model, that is convolutional neural networks model, for the analysis of images, annotating the images data, finding out the layers, how many layers of images, and getting useful information from that for our analysis. Now the last one is the image management. Now it sums up all the techniques that provided the efficient storage, communication, transmission, and retrieval of image data. Telemedicine is an important aspect of image management and formats used to store image that are GIF, that is a 8-bit non-destructive compressed bitmap format, and JPEG, it's a 24-bit destructive compressed bitmap, and TID, well, it's basically a stack of images format. It's a 24-bit publication bitmap. Now image compression is also a part of image management through which a large number of images can be stored and made available through appropriate communication networks and protocols. These are very helpful in research while remotely doing research. Now let's finally come to the conclusion of this presentation on biomedical signal and image processing. There are multiple imaging systems in use: X-ray and digital radiography, magnetic resonance imaging systems, ultrasonic imaging system, X-ray, computed tomography, and more. They have enabled for accurate and fast quantitative analysis and visualization of medical images. Now, these technologies have enabled research scientists and doctors to remote sites to easily share medical data and analyze, thereby enhancing their ability to diagnose, monitor, and treat numerous medical disorders efficiently before it becomes very dangerous for a patient. Now, due to the advancement in image processing, it has now become possible to acquire high-quality images of human body parts and further analyze them using softwares for early detection of deadly medical conditions. So these are the references that I have used in this presentation. And finally, I would like to thank Dr. Mayank Goswami for providing me the image dataset of the OCT scans. And thank you.